I'm interested in how T cells use alternative splicing, a mechanism of gene regulation, to achieve immune responses. Alternative splicing allows for cells to produce distinct proteins from the same gene. This is achieved by combining different pieces of the coding regions of RNA together. I am especially interested in how, in response to T cell receptor signaling, when T cells encounter an antigen, such as a virus or a bacteria, that signaling leads to alternative splicing of signaling molecules themselves, that in turn allows cells to regulate signaling pathways.
Nicole started off in my lab setting out to do a very unique thing, which was to identify all of the genes that undergo alternative splicing during T cell activation. Because of that, her first paper really was a list of all of these genes that undergo this process that we're very interested in studying. One of those genes, MKK7, has become the focus of her own PhD studies.
What most excites me about this project is that we've been able to identify a pathway that communicates information from the cell surface to intracellular signaling cascade down to a nuclear splicing factor that then changes splicing outcome and impacts immune function.
Nicole is a really special student in that she came in with a great background, and she's incredibly smart, but in addition has something that you can't teach, which is a drive and an interest in science that really allows a student to push past all the barriers and the frustrations and fall in love with the field and really achieve great things.
The reason I chose to work on Kristen's lab, well, one was scientifically. I had taken an immunology course in grad school and sort of became interested in immunology, and I had done a rotation in an immunology lab, but my background was biochemistry. So then when I met Kristen, she had a lab where she was asking questions about immune biology, but from an RNA biochemistry perspective. And that really sort of seemed like an exciting angle to go out at, something that meshed well with my background.
Students are great because they bring in their own perspective and their own expertise and their own interests.
From the beginning, it was clear that she was the type of mentor that would foster creativity in her students and would allow them to become independent and grow as scientists.
My own interests shape the lab to some extent, but I'm only one person and I'm limited, so having other people's points of view, other people's interests and biases really broaden the whole research enterprise and enrich the sort of science that we do.
Kristen has impacted my career in many ways. First of all, she made me fall in love with RNA, something that I hope to continue on and carry with me to my future studies. She's been an excellent role model for a well-balanced, effective academic researcher, which is something that I aspire to be. She's been a great supporter, offering me advice and being my best advocate. So I hope to be a mentor, as good of a mentor as she's been to me, to my own students in the future. And I guess the other thing is that she has a passion for science and she has a very practical approach to doing research, which I think has made her so successful. And I hope that I can imitate that in the future.
I think one of the great strengths of doing a PhD at Penn is the integration of many different fields here on campus. Unlike many schools, we have biochemistry, chemistry, physics, and all disciplines of biomedical science all within one campus. And that allows for much more interdisciplinary work than might be possible at other institutions.
I interviewed several places, but I remember coming to Penn and just knowing it sort of clicked. Everyone I interacted with was very friendly and encouraging. It was a collegial environment, not competitive between colleagues, and the students got along well, and the research was cutting edge and exciting, so I just knew that it was the right fit for me.